Hi class, welcome to our last unit for the year, Unit 8, where we will be talking about RNA and protein synthesis and basically what happens to all that DNA. What's the point of all that DNA? We know that DNA is the basic code of life, so how do we use it? And so this is your first video, and today we'll be talking about RNA. So before we get started, let's talk about your learning targets. So today's video will help you compare and contrast RNA and DNA, and will help explain the process of transcription. So by the end of this video, you should be able to compare and contrast RNA and DNA, and you should be able to explain the general process of transcription. So you should have a very good understanding of DNA after our previous unit. And so today we'll talk a little bit about RNA and how DNA kind of becomes or plays a role with RNA. All right, so our big idea for this unit is information and heredity, similar to that of our last unit. And our unit question is, how does information flow from the cell nucleus to direct the synthesis of proteins in the cytoplasm? So DNA doesn't actually do that much for you. I mean, it does a lot. But what it really does is hold the instructions to make the proteins that make up who you are. A lot of your body is primarily composed of proteins, and if you think about it, it makes sense. You can think about your hair is protein, your nails are protein, your muscles have protein. All of that is protein-based, right? Enzymes, all of those types of proteins, biological macromolecules that make up you. And those proteins get made from RNA, which is made from DNA. And so we're going to talk about this transition and what we call the central dogma of molecular biology. Today, though, we're just going to answer the question, what is RNA? We're going to start with comparing and contrasting RNA and DNA. So first, before we go on, I want you to write down everything you think you know about RNA based on what we've talked about and what you've heard or what you can infer. So if you want to take a moment, you can pause the video and then fill in the spaces on your note-taking outline. Write down everything you know about RNA. Don't leave it blank. At least a couple of things. We have talked about RNA a little bit because it is a nucleic acid, but maybe you don't know a ton about it. So that's okay. Just write down what you know. Okay, so let's start by talking about kind of the role of RNA and what RNA does in our body. DNA doesn't actually explain how a gene works. So DNA codes for all the genes. It has all the instructions to make the genes, but DNA isn't actually what turns the genes on or off or makes them function. Before they can be functional, they have to go through RNA and become proteins. That's where RNA comes in. So the answer came from ribonucleic acid, which is another type of nucleic acid. So remember, we have deoxyribonucleic acid, which is our DNA, and now we have ribonucleic acid, which is our RNA. And there are some structural differences between DNA and RNA, as you can probably already tell from the image. For starters, it's single-stranded, so we've got that. We also have a couple of base substitutions. Notice that there's no thymine in RNA, but instead we have uracil, so you've got that going on. And there's also the difference between the sugar. With DNA, you have the deoxyribose sugar, and with RNA, you have a ribose sugar. So those are some basic structural differences between DNA and RNA. So RNA, like DNA, is a nucleic acid that consists of a long chain of nucleotides. Remember, those nucleotides are important. In general, genes contain coded DNA instructions that tell cells how to build proteins. And it's proteins that actually give you the functionality of whatever it is you as an organism are doing. But the first step in decoding or reading these instructions is to copy part of the DNA into RNA. So you can think about this sort of like building a model Lego set or something. If you get a box of Legos and you're going to build something, it's going to come with a bunch of pieces to build it. It's going to come with an instruction booklet, and then it's going to have the box. So if you want to think of the genes as kind of like the little Lego pieces, DNA is like the instruction booklet. And so DNA is the booklet that tells you how to build the thing, but it still needs to be built, and that's where RNA comes into play. Okay, so let's talk a little bit about how this process happens, and we'll go into the details a little bit later because it is a bit of a complex process. RNA uses the instructions that DNA has to direct the building of proteins. Proteins are actually the functional aspect of you. So if you take a look here, we have our DNA tightly and safely packed away in the nucleus. DNA doesn't actually ever leave the nucleus. That would be a no-no. That would be a bad thing. So instead, we have RNA. But what happens is this DNA has to get copied. We call this transcription into RNA. We'll call this mRNA. And we'll talk a little bit more about the different types of RNA in a little bit. And then finally, that RNA will get translated and will build a protein. And so if you don't really know what any of these things are, don't worry about it. We haven't talked about it just yet. This is the general overview. And so we'll dive a little bit into what each of these steps are. Okay, so let's get to the comparing and contrasting of DNA and RNA. And I've actually already talked about this a little bit. They're very similar in structure, which is important because if you're going to copy a segment of DNA, you have to have some similarities in its structure. Whoops, sorry about that. So, reminder, it's single-stranded versus double-stranded. We've got our RNA over here and we've got our DNA over here. So RNA single-stranded, DNA double-stranded. Both of them have cytosine, both of them have guanine, both of them have adenine, but RNA has uracil and DNA has thymine, and so that's a little bit different there. And don't forget that their sugars are different. Remember that DNA has a deoxyribose sugar and RNA has a ribose sugar, and so that's a little bit different. Okay, so those are the main structural differences. A lot of similarities, but a couple of differences as well. So cells use what we call the DNA master plan to construct RNA blueprints that can be used to build proteins. And this makes sense because if you have a master plan, if you make any modification to the master plan, and you're trying to, let's say you're trying to build a skyscraper, right? The master plan is how you're going to build the skyscraper. If you make any change to the master plan, you're going to completely change the skyscraper. So the master plan is usually kept safe somewhere in an office building. However, we have to make copies of the master plan so we can distribute it to the architects, to everybody who's going to play a role in the building of the skyscraper. So we take the master plan and we make copies of it, and then we distribute it to all the people who might need the instructions for building the skyscraper. So the master plan is the DNA. It stays safe and tucks away in the nucleus. It doesn't leave the nucleus because any change to the DNA is going to change the copies, which is going to change the protein. RNA is like the blueprint. It's the copy of the master plan. We can make many copies of RNA and distribute it everywhere it needs to go in order to make as many proteins as we could ever want. So let's talk a little bit more about the functions of RNA then. RNA has lots of different functions, but most only do one job, and that primary job is to build proteins. Most RNA always functions in the building of proteins. So RNA controls the assembly of amino acids into proteins, and we'll talk a little bit more about that. Remember our amino acids that we talked about way back when, at the beginning of the year when we were talking about our biological macromolecules. Chains of amino acids make up proteins. RNA basically helps link these amino acids together in order to build bigger proteins. Each type of RNA molecule specializes in a different aspect of this protein building job. And so there are lots of different types of mRNA, but we're really only going to talk about 3 primary different types of RNA. So the 3 main types of RNA are messenger RNA, and messenger RNA does exactly what it says it's going to do. It is the messenger. It carries the copied DNA to the ribosome so that the message gets received properly. Then we have ribosomal RNA. Remember that the cell has many different ribosomes. Some are free-floating, some are bound. Well, those ribosomes are made up of ribosomal RNA. Remember that ribosomes have 2 subunits, a big subunit and a small subunit. There can be over 80 different types of ribosomal RNA. And so the ribosome is actually where protein synthesis takes place. And so it, in and of itself, has its own special type of RNA. Then finally, we have the transfer RNA. And the transfer RNA actually plays the role of attaching the amino acids in the right sequence that was sent by the messenger RNA. Okay, so that is a snapshot of the different types of RNA in your body. Now there are more, but these are the three we're going to focus on. And these are the three that play a major role in protein synthesis. So let's talk a little bit more about messenger RNA first. Messenger RNA carries the instructions for polypeptide synthesis from the nucleus to the ribosomes in the cytoplasm. We'll talk about how messenger RNA is made in just a minute. That's the actual transcription process. But the important part is it is the blueprint, right? It's going to bring the instructions from the nucleus to the ribosome, and then it's going to allow the other types of RNA to do their job. Ribosomal RNA, as we talked about, is abbreviated rRNA. It forms an important part of both subunits of the ribosomes. Remember, ribosomes can be bound or free-floating, which means they're on the endoplasmic reticulum or they're going to be found in the cytoplasm. Remember that they're made up of two subunits. We have the large subunit, which in this case is the blue blob, and then we have the small subunit, which in this case is the yellow blob. The subunits are made up of several rRNA molecules and as many as 80 different types of proteins in and of themselves. So this just shows you the importance of proteins. Before you can even have a ribosome, you have to build a protein. And so we'll talk more about building proteins in a minute. Finally, we have tRNA or transfer RNA. So after a protein is built, transfer RNA transfers each amino acid to the ribosome as it is specified by the coded messages in the mRNA. And this is a little bit deceiving because it's not necessarily after the protein is built, but it is the actual process of building the protein. That's where the transfer RNA comes in. It's the process of building the protein. These amino acids up here, indicated by this little green ball, get linked together to build the overall polypeptide that is the protein. And we'll talk a little bit more about these, but it has to do with what we call this code. And this little sequence is what's going to allow the amino acid to go in the right location. But again, more on that in a little bit. So it carries the amino acid to the ribosome and then matches them to the coded mRNA message. So now we've talked about the three major types of RNA: mRNA, messenger RNA, rRNA, ribosomal RNA, and tRNA, or transfer RNA, all of which play an important role in the protein synthesis. So now we're going to completely transition and talk about the transcription portion. Because before we can talk about protein synthesis, we have to talk about RNA synthesis, where that is the making of RNA. Remember that to synthesize something means to make something. So before we can do anything, we actually have to copy the DNA into RNA, and that's what we call transcription. Most of the work of making RNA takes place during transcription. Transcription is when segments of DNA serve as templates to produce complementary RNA molecules. You can think of it like this. If I didn't have a copy machine and I wanted you to copy a letter for me, I would give you a letter and you would sit there and by hand you would write it out letter by letter, word for word until you had copied the exact letter. So the same is true for the idea of going from DNA to RNA. So the bases of the translated RNA component complement that of the DNA. And that's why those bases, those nitrogenous bases, adenine, thymine, guanine, and cytosine are going to be so important. And we'll talk about why there's a little bit of a difference in the bases of RNA and DNA. All right. So in prokaryotes, RNA synthesis and protein synthesis take place in the cytoplasm because remember, prokaryotes don't have a nucleus. And so it's a little bit, the process of RNA synthesis is a little bit more simple, less complex, right? But prokaryotes in general are less complex. However, in eukaryotes, RNA synthesis has to take place in the nucleus because we don't ever take the DNA out of the nucleus. And then the RNA is moved to the cytoplasm with the help of proteins. And we'll talk more about how that happens in a second. So if you look at this picture, this kind of gives you an overall snapshot of the protein synthesis process. So transcription takes place in the nucleus. The shell's machinery copies the gene sequence into mRNA, or messenger RNA. And this molecule is similar to DNA, and like DNA, mRNA has 4 nucleotide bases. But remember, we don't have a uracil. We don't have a thymine. We have a uracil when we're talking about RNA. Then, after we have transcribed the DNA into RNA, that mRNA is going to be finalized, and then it's going to leave the nucleus, where it can experience what we'll call translation. And we'll talk about translation in our next lesson. And translation is where the actual protein gets processed. So I'm going to skip translation for now because you'll learn about it later. But let's talk more about transcription, because that's what we need to know before we can talk about translation. Transcription requires an enzyme known as RNA polymerase. Now, what does RNA polymerase remind you of? Probably the enzyme that helps with the replication of DNA, DNA polymerase. Well, this makes sense because RNA polymerase does a lot of the same things that DNA polymerase does. However, it's specific to RNA. RNA polymerase binds to the DNA during transcription and separates out the DNA strands, then attaches the bases onto RNA. So here you can see an image of what this process looks like. Gray blob is the RNA polymerase. Notice how it separates the DNA here. So you have one strand of DNA up here, and then you have one strand of DNA down here. And then you have the bases attaching to build a single-stranded RNA molecule, right? So just keep that in mind. Now, RNA polymerase then uses one strand of the DNA as a template, and then it can assemble a complementary strand of RNA. Now, I want you to think a little bit abstractly about this. So remember that you have your bases A, T, C, G, and they have all these different combinations, and all these different combinations code for genes. Well, they also can be copied, and the RNA can code for these same genes. And so again, something we'll talk about later, but I want you to be thinking about it as we go on. So, how does RNA know where to start and stop? Because we're not going to copy an entire DNA molecule all in one. In fact, we're only going to transcribe the piece of DNA that we need. If we need one gene, we're going to transcribe that one section. So we're not going to transcribe an entire DNA molecule. That's a waste of energy. It's not very energy efficient. So how does RNA know where on the DNA to start and where on the DNA to stop if we're not copying the whole thing? Well, RNA, or DNA and RNA have these things called promoters. They promote transcription. So RNA polymerase doesn't bind anywhere. It can only bind to a promoter. So if you have a long strand of DNA, your RNA can only bind to the spot that has the promoter. You can think of the promoter like a green light, which means go, or a starting point. Promoters are regions of DNA that have specific base pair sequences. Remember, our bases are A, T, C, and G. And if there are stop or start sequences, then there are also stop sequences. So if you have a green light, which means go, you're also going to have a red light, which means stop. So RNA synthesis does have a specific point in which it needs to start and a specific point in which it needs to stop. So I have examples of both stop and start sequences. So the example of a stop sequence is UAA. Now, one thing that you're going to learn is that all of these come in what are called triplets, or 3 bases at once. The triplet sequence for stop is uracil, adenine, and adenine. That's abbreviated UAA. When an RNA polymerase sees UAA as it's making its copies, it's going to stop. You can think of it like a period. If you see a period and you're transcribing my letter and you see a period, you know you're going to stop. An example of a start sequence is AUG, adenine, uracil, and guanine. When RNA sees this AUG, it's going to start. It's going to say, oh, this is where I go. So it's going to start at AUG and it's going to stop at UAA. So it might look something like this. Your RNA polymerase, you bind here, and you'd copy, and you'd copy, and you'd copy, and then you'd stop here. And then you would take your single-stranded segment of RNA out of the nucleus and off to the ribosome where you will make a protein. So promoters are signals in the DNA molecule that show RNA polymerase exactly where to start making RNA. So I want you to notice something though. Remember that we've got some substitutions. So what is the base that DNA has and the base that RNA has that are different? Remember that we have thymine and uracil. We're going to make the DNA version of this. Remember that your A is going to correspond to, that your A is going to correspond to T, your U is going to correspond to A, your G is going to correspond to C, T, and then we've got G, U, and so on and so forth. So remember, and this is something we'll talk about more in our next unit, that we're talking about copying from DNA to RNA. So just something to keep in mind. So before anything else can happen, before this mRNA can actually leave the nucleus, it needs to get edited. So we've gone through the process. We've gone ahead and split up our DNA. We've made a copy of our DNA, but that's the rough draft. Just like if you're transcribing a poem for me or a letter, you wouldn't copy it down real fast and be like, okay, done. You would go through and you'd proofread it and you'd say, all right, did I make a typo? Did I spell everything correctly? Did I miss a word? So your body does the same thing, and it's called RNA editing. Portions of RNA actually get cut out and removed from the molecule before it is completed. And the things that get removed are introns or exons. So some things stay in and some things go out. Introns get cut out and the exons get smushed together. So introns are portions that are cut out and thrown away, and exons are the leftover pieces that were not removed, and they get spliced together to form the final RNA molecule. Now, there are a lot of different reasons why these introns potentially get cut out. Scientists are actually still trying to learn the purpose of some of this RNA editing. Some people speculate that technically introns and exons can vary, and so depending on which introns and which exons you cut out and leave in determines which genes are actually turned on and off. But the truth is we don't actually know a ton about why RNA editing happens, just that it does. So here is kind of what the process might look like. You've got your DNA up here, that's this blue one. Then you've made your RNA, and we're not going to talk too much about the cap and the tail, but the cap and the tail are kind of like safety features that prevent the RNA from getting damaged or unraveling. And then what happens is these introns get removed, exons get spliced together, and now you have the coding sequence that is ready to leave the nucleus, go into the cytoplasm, where it will find its ribosome. Now, you've learned a lot today. So I want you to take a minute and answer this question. What is RNA? And I want you to answer it in as much detail as possible. Be specific, because how much do you already know? And the more you know, the more you're going to take into the next lesson. And also, don't forget before you go to assess your learning targets. Can you compare and contrast RNA and DNA? Do you have a really good understanding of the difference between the two of them? And can you explain the process of transcription? This one's a little more complicated. In general, remember that transcription is the process of going from DNA to RNA. So can you explain in general how that happens? So if you can't, I would encourage you to watch the video again. Don't forget to read the chapter, and then stay up on all your homework this week. Otherwise, it's been great, and I'll see you guys next week. Bye.